Microbial communities are among the main driving forces of biogeochemical processes in the biosphere. Standard approaches to studying the connection between microbial communities and biological phenotypes relies on abundance matrices to represent the microbial compositions. However, these methods have limitations in finding real connections between microbes and phenotypes. These methods often focus on the relationship between a single OTU and a phenotype, a univariate measure which has multiple assumptions violated by the complexity of the microbiome, leading to elevated type 1 error and reduced power. One solution to this is Bayesian network regression. This model was originally developed on brain connectome data by Guhan Rodriguez in 2020. The original model was designed to determine influential brain regions and the connections between them. It is very well performing in this application. However, brain connectome data is completely dense, whereas microbiome networks are quite sparse. In our work, we aim to extend this model and implement it in the Julia programming language. We evaluate performance on simulated microbiome data, identifying influential microbes and the connections between them in some plant response, and then analyze phosphorus leaching and soil microbiome data collected by Wag et al. In 2019. The model is quite simple. The response is seen as the product of an adjacency matrix A, describing the microbial network, and B, a coefficient matrix, describing the relationship between the network and the response, plus some random noise. We're able to get maximum a posteriori estimates of B, as well as estimates of the probability that each microbe is influential on the response. We performed two sets of simulations. In theoretical simulations, the response is generated directly from the model itself. In realistic simulations, we look at three separate models. The first, the response is generated just based on whether the microbe is in a sample or is not in a sample. In the interaction model, we add to this a product of the adjacency matrix and the coefficient matrix. So it looks at both presence, absence of the microbe, as well as the relationships between microbes. And finally, we look at a functional redundancy model where this interaction model is capped at a specific value. We find good results in the functional redundancy model at high sizes. And as you can see, the model suffers from low power when the sample size is small. Finally, we apply the model to soil microbiome and phosphorus leaching data from Wag et al. We apply data augmentation techniques to increase the sample size from 50 to 200 in order to increase the power. Posterior probability of influence measures are unable to identify any influential microbes. However, using 90% credible intervals of microbe coefficients, we can identify two influential microbes, one of which was not identified by the original Wag et al. Study. For more information, please read our paper in Ecology and Evolution. And finally, thank you both to the SolisLemus-lab and to the Department of Energy for funding.